we will have a discussion on the structure of mitochondria. So mitochondria are of different shapes seen in different types of cells, and usually they are rod-like cell organelles. Mitochondria are present in virtually all the eukaryotic cells as well as the plastid seen in plant cells. The chloroplasts etc. contain mitochondria inside, and they are double membrane bound organelles which are bounded by two membranes, and externally they look like elongated cylinders ranging from a diameter of 0.5 to 1 microns, and they are considered to be the aerobic respiration centers of the cell. So what is aerobic respiration? Respiration is the process, or it is the oxidation by which nutrients are broken down in cells to release their energy. For example, glucose is the primary fuel of our cells, and glucose is to be oxidized so as to harness the energy from this molecule, and this energy is used for carrying out the life processes of the cell. So the glucose is first broken down to pyruvic acid in a series of reactions which is called as glycolysis, and that can take place in the absence of oxygen. But this pyruvic acid, what is the fate of pyruvic acid? If there is enough oxygen, if the cell is in an aerobic condition, this pyruvic acid will enter into mitochondria, where it will be converted into acetyl coenzyme A. This acetyl coenzyme A will enter into the Krebs cycle, and thereafter the products of Krebs cycle, they will enter into the electron transport chain, and finally there will be synthesis of ATP from ADP. So there will be a number of oxidation reduction reactions or redox reactions taking place in the mitochondria, at the end of which ADP will be phosphorylated to ATP. So it is called as oxidative phosphorylation, in which ATP is synthesized. So where is this ATP coming from? It is the ATP that is synthesized by the phosphorylation of ADP, and this is actually an energy conserving process. That means the energy derived by the oxidation of glucose is stored in the form of ATP. All these reactions take place in the mitochondria, and thereby it is called as a powerhouse of the cell. So it is a center of aerobic respiration because this takes place in the presence of oxygen. But if oxygen is not available, this pyruvic acid cannot enter into the Krebs cycle. This pyruvic acid will be either converted to lactic acid or ethyl alcohol, depending upon the cell type. So for completely tapping the energy from a fuel molecule, it has to enter at some point into the mitochondria, where it will be subjected to Krebs cycle and oxidative phosphorylation. So that is why it is considered to be the powerhouse of the cell, where the energy currency of the cell is made, which is called as ATP. And regarding the distribution of mitochondria, they are seen more in energy demanding cells. For example, the muscles, they have got a region called as the I band, where there will be numerous mitochondria. The axoneme of the sperm tail, which helps in propulsion of the sperm, it will be rich in mitochondria. And they are also seen attached to the endoplasmic reticulum in protein synthesizing cells. But they are absent in prokaryotic cells, mature mammalian RBC, and some non-photosynthetic algae. Now the number of mitochondria, that varies depending upon the cell type. In sperms, certain sperms, there will be only one mitochondrion. In flagellate protozoans like Chromulina, Trypanosoma, and some algae like Micromonas, there will be one mitochondria per cell. The number can be 1000 to 1600 in mammalian liver cells. It can be 10 lakh, it can be 1 lakh in the giant amoeba called as Pelomyxa. It can be 15 lakhs in sea urchin eggs. It can be 30 lakh in oocytes, and it is abundant in the insect flight muscles. So all these are, depending upon the energy requirement, the distribution of mitochondria will be different. Here we can see the sperm cells, they require propulsion energy, for which they are richly supplied with mitochondria. The flight muscles of insects and birds are continuously contracting and relaxing to enable flight movements. They are having plenty of mitochondria. The meristematic cells of plants, they contain large amounts of mitochondria. Regarding the studies on mitochondria, they were first observed by Kolliker in 1880. Thereafter, in 1894, they were named as bioblasts by Richard Altman. In 1897, C. Benda named them mitochondria. In the late 1890s, Michaelis suggested that they are the centers of redox reaction in the cell, and this hypothesis was confirmed by Kingberry in 1912. And in late 1940s and 50s, lot of studies were conducted on mitochondria regarding their isolation, fixation and sectioning, and they were conducted by Claude, Palade and Potter. Coming to the oxidative phosphorylation mitochondria, when the glucose is converted to pyruvate by glycolysis, and if it has to go to either lactic acid or ethyl alcohol through fermentation in the absence of oxygen, only less amount of energy is tapped from glucose. But if there is oxygen, this pyruvic acid can enter into mitochondria, where complete oxidation of glucose takes place into carbon dioxide and water. So, compared to the energy released by glycolysis, the energy released by Krebs cycle and the respiratory chain in the mitochondria is very high when you take any fuel molecule. It's a double membranous organelle with an outer membrane and an inner membrane, and this is both these membranes have the typical constitution of the plasma membrane, that is, it is a lipid bilayer which is having extrinsic and intrinsic proteins, and in between the inner membrane and the outer membrane there is a space which is called as the intermembrane space. So, the space between the two is called as the intermembrane space, and inside the inner membrane there is a matrix. There is a matrix which is fluid filled. So, these are the main components of mitochondria: the outer membrane, the inner membrane, and the space between the outer and the inner membranes, and the matrix inside the mitochondrion, and it also contains ribosomes and other proteins, enzymes, etc., which we will come to. We will have a look on the outer and inner membranes of mitochondria. Though they are lipid bilayers, there are slight differences in the composition. That is, in outer membrane, the lipid to protein ratio is 40:60, whereas in the inner membrane, this ratio is 25:75. So, 40% lipids and 60% proteins constitute the outer membrane, and it contains a number of transport proteins which are called porins. So, they act like channels. They have pores inside through which they will allow substances to pass through. Usually they act like a sieve that allows only small molecules to pass through. That means molecules up to a size of 5000 Daltons or less. And they will cross this outer mitochondrial membrane and enter into the intermembrane space. Further will they go into the mitochondrial matrix, that is determined by the permeability of the inner membrane. Similar and likewise, the outer membrane also contains larger transport proteins which are called translocases, which will also help in allowing larger molecules to pass through. The inner membrane is the center for oxidative phosphorylation and ATP synthesis. So, all these reactions are concentrated on the inner membrane, and these translocases, they allow the transport of substances like aspartate, glutamate, phosphate, ADP, ATP, etc. So, all these substances will be allowed to pass through the inner membrane, and thereby they can reach the mitochondrial matrix. So, they are all taking part in the oxidation reduction reactions and other metabolic reactions inside the mitochondria. And another characteristic of inner membrane is the presence of a double phospholipid which is called as cardiolipin. This is also there in the outer membrane, but its concentration is high in the inner membrane. So, cardiolipin is characteristic of the inner membrane of mitochondria, though it is found in low amounts in the outer membrane also. And what is the function of cardiolipin? Cardiolipin makes the membrane impermeable to ions. Then, so it is actually a filter, we can say. It makes the membrane permeable to ions, and it will help in maintaining the structure of certain proteins like cytochrome C oxidase, which is an important participant of the oxidative phosphorylation reaction. So, the quaternary structure of cytochrome C oxidase, etc., will be maintained by cardiolipin, and it also helps in translocation of cholesterol. And apart from this translocases and cardiolipin, the inner membrane also contains a variety of other transport proteins which makes it permeable to some small molecules which can cross and reach the matrix, because there are some chemical reactions taking place in the matrix for which enzymes are required, and these enzyme molecules can travel through the inner membrane of the mitochondrion with the help of these other transport proteins apart from translocases. Now, just go to this diagram. We can see this is the inner membrane and this is the outer membrane. So, this is the outer membrane. So, you can see the space in between. You can see space that is called as the intermembrane space, but at certain regions there are places where they come in close opposition with each other. There are places where they come in contact. They are called as the contact sites. And they play a prominent role in the transport of certain proteins into the mitochondria, certain proteins which are encoded by genes in the nucleus, but they have to come into their function takes place in the mitochondria. Even though these proteins are synthesized in the cytoplasm, they are functional inside the mitochondria, so they are to be transported in, and these contact sites, they help in the transport of such precursor proteins into the mitochondria to carry out their further functions. And you can also see in the contact zones there will be large amounts of cytoribosomes also. The cytoribosomes are also abundant or seen crowded in the contact sites between the inner and the outer membrane. The inner mitochondrial membrane harbors the enzymes which take part in the respiratory chain. So, they are essential for the process of oxidative phosphorylation. Then only ATP will be synthesized. And for this, the inner membrane has to have an increased surface area because it is lodging the enzymes of the respiratory chain. So, for this purpose, the inner membrane is highly convoluted and it forms a series of infoldings, and these infoldings are called as cristae. So, we can see that the cristae are folded into the matrix. So, these are the cristae which are infoldings of the inner membrane which project into the mitochondrial matrix. They increase the surface area of the inner membrane. If you take a liver cell, because of the presence of cristae, the area of the inner membrane is tripled. Then the cristae also show regions called as cristae junctions where they are connected to the inner membrane. Here you can see they are opening to the intermembrane space. So, these regions are called as cristae junctions. So, they are also characteristic of mitochondria. The cristae junctions connect the cristae to the inner boundary membrane. And the number of cristae is also will differ depending upon the cell type. In a cardiac muscle cell which requires higher energy than a liver cell, there will be more number of cristae. That means the number of cristae is 3 times greater in the cardiac muscle cell mitochondrion compared to that of a liver cell. So, the number of cristae also depends upon the energy demand for ATP. Then the shape of cristae, they may be transverse, longitudinal, lamellar or plate-like or sheet-like. Then tubular cristae, so you can see tubular cristae. Then vesicular cristae, they look like sacs. These are the vesicular cristae, and reticulate, that means in the form of a network. So, cristae can be transverse, longitudinal, lamellar or plate-like or flat, tubular, vesicular which is sac-like, and reticulate which is network-like. Mitochondria can assume 2 types of conformations. One is called condensed conformation and the other is called orthodox conformation. So, what is the difference between the two? So, this is the condensed conformation. So, this is a contracted state of mitochondria, whereas orthodox is an expanded state of mitochondria, we can say. In condensed state, there will be enough space between the outer membrane and the inner membrane. That means the outer membrane and inner membrane are having greater space in between in the condensed state, whereas in orthodox state the distance between the outer membrane and inner membrane will be small. So the intermembrane space will be smaller in the orthodox state. Then in the condensed state, because of the condensation or the contraction of the inner membrane towards the matrix, the cristae will be plate-like or lamellar. Cristae will be lamellar. Whereas in orthodox state, the cristae will be almost tubular. They will be somewhat tubular. And this condensed state, you can say it is the active state, and the orthodox state is usually seen in times where there is low respiration taking place. So these are the differences between the condensed and orthodox states of mitochondria. Condensed is a contracted state with swollen intermembrane space and lamellar cristae, and they will be having a very dense matrix and wider cristae, whereas in orthodox state you can see an expanded less dense matrix with tubular cristae, and the cristae compartments will be more compact. They will have a larger matrix volume, and they are seen in low respiration states. So here also you can see the difference between a condensed state and an orthodox state. The cristae and the inner membrane are characterized by the presence of certain stalked particles which are called by different names. They are called F0, F1 particles, elementary particles, oxysomes, Fernandez-Moran subunits, ATP synthesis, mitochondrial ATPases, etc. So studies were conducted on their structure as well as the role played by them, and for their study Paul Boyer and John Walker were awarded the Nobel Prize in Chemistry in 1997. Now coming to the structure of these particles, they are transmembrane proteins which are seen spanning the inner membrane, and they have got three parts: the base piece or the F0 unit, the stalk or the F5, F6 particle, and the head or the F1 particle. The base piece is located in the inner membrane and it is hydrophobic, and it is formed of polypeptides, proteolipids, etc., and they provide channels for protons. Then the middle stalk, it contains characteristic proteins which are called OSCP or oligomycin sensitivity conferring proteins, which are characteristic of this region of F0, F1 particle. And the head region or the F1 particle is hydrophilic. It is projecting into the matrix and it is capped with ATPase inhibitor protein. So this is the base, this is the stalk, and this is the head. So we will just go to the head region. The head region in turn has got 9 subunits. Which are they? You can see 1, 2, 3. 3 alpha, 1, 2, 3 beta, then 1 gamma, 1 epsilon, and 1 delta. So these 9 subunits constitute the F1 particle or the head. So this is how it is arranged. So these are the subunits, and here you can see the base is embedded in the lipid bilayer, and you can see the stalk. Then the enzymes of the inner membrane which take part in oxidative phosphorylation and electron transport chain, all these enzymes can be grouped into 5. They are actually remaining as 5 different complexes. All these enzymes are categorized into 5 different complexes. They are complex 1, 2, 3, 4 and 5. And this complex 1 and 2 are located in the base region. So the enzyme complexes 1 and 2 are located in the base region. The complex 3 is there in the stalk region. Complex 4 and 5 in the head region. So complexes 1 and 2 in base piece, complex 3 in the stalk, complex 4 and 5 in the head region. Now complexes from 1 to 4 are constituents of respiratory chain. That is, the enzymes present in complexes 1 to 4 are those enzymes which take part in the respiratory chain. Whereas this complex 5 consists of enzymes which are taking part in ATP synthesis. And you can also see two electron carriers. There are three electron carriers, the coenzyme Q, cytochrome C and ubiquinone. When all these enzyme complexes are, you can say, somewhat stable, there are mobile electron carriers also. These mobile electron carriers are, one is coenzyme Q. It is a mobile electron carrier that operates between 1 and 3 as well as 2 and 3. There is another mobile electron carrier which is called cytochrome C. It is a mobile electron carrier that acts between 3 and 4. So, apart from the 5 enzyme complexes, there are mobile electron carriers also, which are coenzyme Q, cytochrome C, etc. And coenzyme Q operates between 1 and 3 as well as 2 and 3. Cytochrome C operates between 3 and 4. And here also you can say all these take part in the respiratory chain, whereas the last one, complex 5, takes part in ATP synthesis. Now the matrix. The matrix is dense, gel-like, protein-rich. And the two main constituents of the matrix are, one, the enzymes responsible for oxidative metabolism. Second one, the mitochondrial genetic system. So these two are the main constituents of the matrix of mitochondria. We go to the oxidative metabolism. As I already told you, the oxidative breakdown of glucose, fatty acids, etc. is the principal energy source for any cell. So it is the source of metabolic energy. And when we take the case of glucose, it is first broken down to pyruvic acid in the cytoplasm by an anaerobic method called as glycolysis. This pyruvate is then transported to the mitochondria, where it enters into a cycle of reactions which is called as Krebs cycle or citric acid cycle. So before that, this pyruvic acid is converted to acetyl coenzyme A by an oxidation reaction. And this pyruvic acid, once oxidized to acetyl coenzyme A, will enter into the Krebs cycle and it is broken down to carbon dioxide. Now, coming to the fatty acids. The fatty acids are also oxidized, and that takes place in the mitochondria. The oxidation of fatty acids will also yield acetyl coenzyme A, which will again enter the citric acid cycle. So, acetyl coenzyme A will be derived from glucose through pyruvate as well as by the oxidation of fatty acids. Both these yield acetyl coenzyme A, which will enter the citric acid cycle, and that takes place in the mitochondria. So the enzymes of the citric acid cycle are located in the matrix of mitochondria, and they are the central players in the oxidative metabolism of carbohydrates and fat. So this is the citric acid cycle in a nutshell, where I have to tell only one thing, that is, there is one enzyme of the citric acid cycle which is called succinate dehydrogenase. Only this enzyme is located on the inner membrane. All the other enzymes which take part in citric acid cycle are located in the matrix, whereas only this enzyme is located in the inner membrane. You can see succinyl coenzyme A synthetase, alpha-ketoglutarate dehydrogenase, isocitrate dehydrogenase, aconitase, malate dehydrogenase, fumarase. All these are located in the matrix except the succinate dehydrogenase, which is located in the inner membrane. Then the mitochondrial DNA. There is genetic material inside the mitochondria also. You know that the genetic material is there in the nucleus, but the mitochondria also contains genetic material which is called as the mitochondrial DNA, but it only represents a small fraction of the total DNA in the cells. And just like nuclear DNA, it is also double-stranded, but it is circular and covalently closed, whereas our nuclear DNA is linear. But mitochondrial DNA is covalently closed and circular, but it is present in multiple copies. So you can see the mitochondrial DNA, and they are seen in the matrix, and sometimes they will be seen attached to the inner mitochondrial membrane. Then the mitochondrial DNA has got around 16,500 base pairs, and they all constitute 37 genes. And out of these 37 genes, 13 are used for oxidative phosphorylation, and the remaining are used for making tRNA, rRNA, etc. and the tRNA and rRNA which are there inside the mitochondria. So out of 37 genes, 13 give gene products which take part in oxidative phosphorylation, and the others code for mitochondrial tRNA and ribosomal RNA, etc. Then the mitochondrial DNA is self-duplicating. That means its replication is independent of the replication of chromosomal DNA. But for replication, there are certain factors required, replication factors. These factors are coded by genes which are there in nuclear DNA. So we cannot say it is completely independent of nuclear DNA. Its replication is independent of the replication of nuclear DNA, but for replication it requires certain replication factors that will be coded by genes in the nuclear DNA. And the proteins which are required for replication, they are also synthesized in the cytoplasm, in the cytoribosomes, and they come to the mitochondrial matrix, and then the mitochondrial DNA will start duplication. So it is not in synchrony with the replication of the nuclear DNA, that's all. But for that process to take place, it has to depend upon the gene products from the nuclear DNA. And there is one peculiarity. When fertilization takes place, the ovum is fertilized by the sperm, but only the nucleus of the sperm goes to the ovum. The cytoplasm of the ovum only reaches the zygote. So if a zygote inherits DNA from both the parents, nuclear DNA from both the parents, mitochondrial DNA is inherited only from the mother, we can say, because only the egg cytoplasm contributes to the zygote. The sperm cytoplasm does not contribute to the zygote. So the egg cytoplasm contains mitochondria, they contain the mitochondrial genes, and so a zygote will receive mitochondrial DNA only from the mother. So mitochondrial DNA, unlike nuclear DNA, is inherited from the mother, while nuclear DNA is inherited from both the parents. And lastly, the other matrix components are mitochondrial granules, phospholipids, yolk bodies, glycogen deposits, insoluble inorganic salts, etc. So these are the various components of mitochondria.